This video is for The Living Earth, Unit 4.3, Transcription and Translation. Today's essential question: how do cells control gene expression? So today we are going to explore one of the most fascinating and important concepts in biology: gene expression. Imagine that every cell in your body has the exact same DNA, like kind of having the same instruction manual. But not every cell uses the same instructions. For example, your muscle cells and your skin cells have the same DNA, but they look and act completely differently. The question is, why? Because they express different genes. Gene expression is the process that takes the genetic code that's stored in your DNA and uses it to make proteins. And proteins are the molecules that do most of the work in our bodies. For example, they make our traits, they run our chemistry, and even control what cells do and when. Today, we'll learn about how this process works and why it's important. So let's start by defining gene expression a little more clearly. So inside every cell is DNA, and this DNA contains thousands of genes, each one a set of instructions for making a particular protein. But here's the trick: not every gene is turned on or expressed in every cell at all times. It turns out that cells turn certain genes on or off depending on what they need to do. For example, liver cells express genes that help break down toxins. Skin cells express genes that make proteins like keratin for structure. That's why you don't grow hair in your liver. Gene expression is what allows cells to specialize and take on different roles, even though they all have the same DNA. There's a big idea in biology, which we call the central dogma. It explains how information flows in a cell to make traits. It goes like this: DNA makes RNA, and RNA makes protein. This process has two major steps, of which you've already learned: transcription and translation. Recall that transcription is when a cell makes a copy of a gene in the form of RNA. Translation is when that RNA message is read by a ribosome to make a protein. Proteins are the end products of this information flow. They can be structural, like collagen in your skin, or functional, like enzymes that digest food or carry oxygen in your blood. This flow of information, remember from DNA to RNA to protein, is what gives rise to all the traits that we see. Now let's take a closer look at DNA, the blueprint of life. DNA is made up of two strands twisted together in a shape called a double helix. You can think of it like a spiral staircase. The sides are made of a sugar and phosphate backbone, and the steps are pairs of nitrogen bases. Recall that there are four types of bases in DNA: adenine, thymine, cytosine, and guanine. These bases pair in a way that is very specific. A, adenine, will always pair with T, thymine, and C, cytosine, will always pair with G, guanine. This pairing is what makes DNA so stable and reliable for copying. The sequence of these bases, the A's and T's and C's and G's, is like a code, meaning it tells the cell what proteins to make and in what order. Even tiny changes in the sequence can lead to big changes in traits or even cause genetic disorders. Deoxyribonucleic acid, or DNA, is the molecular basis of heredity in all living organisms. Its unique structure enables it to store, replicate, and transmit genetic information with remarkable fidelity. The complementary nature of DNA's structure is fundamental to its function. During replication, the two strands separate, and each serves as a template for synthesizing a new complementary strand. and this ensures that genetic information is preserved through different cell divisions. This elegant molecular architecture represents one of nature's most sophisticated information storage systems, which is capable of encoding the instructions for building and maintaining all living organisms on Earth. Oh, we skipped ahead too far. Let me do this.
Inside of living cells, the regulation of gene expression can take place at any one of a number of different steps in the production of the gene products, RNA and protein. In the case of transcriptional regulation, for example, the rate at which a gene's DNA instructions are transcribed into RNA is controlled. One of the main ways this happens is through the actions of proteins called transcription factors that attach to the DNA. A simple example of this type of regulation occurs when a gene is turned off by a repressor molecule that binds to its controller region. The repressor prevents the RNA polymerase enzyme from moving along the gene and assembling the subunits needed to make messenger RNA. However, in the presence of inducer molecules, the repressor is removed and the transcription stage of gene expression can occur. In the case of post-transcriptional regulation, certain RNA binding proteins attach to the spliced, reassembled RNA transcript. Some of these gene-regulating proteins influence the RNA's stability or determine where the RNA will be exported outside of the nucleus. With a third kind of regulation, called translational regulation, gene expression is controlled at the level of the ribosomes. In one example of this, certain gene-regulating molecules interfere with how well the large and small ribosomal subunits bind together, and so are able to control the rate of translation of messenger RNA into protein that way. A fourth place that gene expression is regulated is at the post-translational level. This generally involves changing the newly produced protein by breaking it down or making chemical alterations to it that affect how it functions in the cell.
This first step in gene expression is called transcription. This happens inside the cell nucleus, where DNA lives. During transcription, recall, an enzyme called RNA polymerase binds to a section of DNA called the promoter. This is like a start here signal. RNA polymerase then unzips the DNA and starts building a strand of RNA with matching RNA bases to the exposed DNA bases. But here's an important difference. In RNA, thymine is replaced by a base called uracil, represented by a capital U. So, if the DNA has an adenine, an A, then RNA will pair it with uracil, a U, not thymine. The result of transcription is a strand of messenger RNA, or mRNA, which carries the instructions from DNA out of the nucleus and into the cytoplasm. Now let's talk about RNA. Unlike DNA, RNA is a single strand. It can move out of the nucleus and into the cytoplasm where proteins are made. The most important type of RNA for gene expression is mRNA, the messenger RNA, because it carries the instructions from the DNA to the ribosomes where the proteins are assembled. But remember, mRNA isn't the only kind of RNA. tRNA, or transfer RNA, helps bring amino acids to the ribosome. And rRNA, or ribosomal RNA, helps make up the structure of the ribosomes. These different RNAs work together during the next step called translation. Translation is the second major step of gene expression. Translation happens at the ribosomes, which are like protein factories in the cell. The ribosome reads the mRNA in sets of 3 bases at a time. Each set of 3 is called a codon, and each codon matches up with a specific amino acid. Here's where tRNA comes in. tRNA molecules have anti-codons that match up with the mRNA codons. Each tRNA carries an amino acid, and it adds that amino acid to a growing chain of amino acids. This chain becomes a protein. Once the entire mRNA message is read and all of the amino acids are linked, the chain then folds on itself into a specific shape and becomes a functional protein. Now let's zoom out for a moment. Your genome is your complete set of DNA, meaning all of your genes. In humans, the genome is stored in 46 chromosomes, which are in each cell nucleus. You get 23 of these chromosomes from your mom and 23 of these chromosomes from your dad. Even though all humans share about 99.9% of their DNA, that tiny 0.1% difference accounts for everything that makes us unique, from our appearance to our risk of certain diseases. Here's a fun fact. If you stretched out the DNA from just one of your cells, it would be about 2 meters long. But it's all packed into a nucleus that's so small, you'd need a microscope to see it. But not all genes are active all the time. Cells control which genes are turned off or on, depending on what they need. This is called gene expression. And there are many ways that this happens. DNA methylation can silence a gene by adding chemical tags. Transcription factors can help or block RNA polymerase from starting transcription. And RNA editing can remove unwanted sections, which we call introns, or protect the RNA with caps and tails. This regulation is what allows cells to respond to their environment, develop properly, and perform specialized functions. One of the coolest examples of gene expression is during development. Think about a tadpole that's turning into a frog. Both the tadpole and the frog have the same DNA, but very different bodies. These bodies transform, and that transformation happens because different genes are turned on and off at different times. In tadpoles, genes for tails and gills are active. Then as it becomes a frog, these genes turn off, and the genes for legs and lungs turn on. This precise control over gene expression is what allows organisms to grow, develop, and adapt. So in the end, it all starts with the DNA. The sequence of bases in your DNA determines which proteins your cells make. And these proteins shape your traits, like eye color, hair texture, blood type, and even how your body digests food. But your environment plays a role as well. Things like diet, stress, temperature, or exposure to toxins can all influence which genes are expressed. That's why identical twins who share the same DNA can still turn out a little different. So gene expression isn't just about genetics, it's also about how genes interact with the world around us. And at this point, you should be able to generate a full and appropriate response to today's essential question: how do cells control gene expression?